Okay, let's take a look at the concept question. Name 3 ways in which the RNA may be processed after a gene is transcribed in the nucleus. So the first way would be cutting out introns. Fire up the pen, make sure that it's working. 1, removing introns. The second one goes hand in hand with removing introns, splicing exons. And the third, adding the cap and poly A tail. So those would be the 3 ways that the RNA can be processed after the gene's transcribed in the nucleus. This all takes place obviously inside the nucleus. So let's move on and take a look at some other parts of this chapter. So, and you may wonder why some of these things are tied together. Once we have cells active during the development of organism, we need a way in order to be able to signal to different cells what they ought to become. Otherwise, it just is a bunch of identical cells. I mean, remember, every cell in your body contains exactly the same set of chromosomes containing exactly the same set of genes. We now know how to regulate the expression of genes through that series of regulatory mechanisms that we've just been talking about. But why do some cells turn on one set of genes, whereas other cells turn on a completely different set of genes and become different types of cells? How do cells know which genes to turn on and which genes to turn off in order to become the type of cell that they become? This is a fairly complex topic, and we're really only just starting to come to terms with the beginnings of how this works. So we're really just very early on in understanding how this takes place. Yet we have a reasonable understanding of what is involved. It turns out that there are signal molecules. Many of them are proteins, but not all of them necessarily have to be proteins. Some of them are hormones. But these signal molecules are released by different cells, circulate through the developing embryo, and bind to target cells, because those target cells have a protein receptor on the surface of the cell that binds the particular signal molecule. The presence of that signal molecule attached to the receptor molecule then turns on sets of genes inside the nucleus. And it is that the presence of the signaling molecule that actually activates the genes in the target cell and ultimately leads to that target cell turning on the set of genes that are going to result in that cell becoming what it becomes. So having an understanding of how this pathway, which is referred to as the signal transduction pathway, actually works is kind of important, because it gives us the clue now as to the connection between regulating the expression of genes in cells and how different types of cells become the cells that they become. It's through the reception of these signals and the effect on transcription in the nucleus that is the key to understanding how these cells can become different types of cells. So here is an overall view of the signaling pathway. The signaling molecule is produced by the cell that is going to release the signal, the signaling cell, which could be a cell producing hormones, or it could be a cell producing protein molecules. but that signal molecule is released out of the signaling cell into the environment. It then binds to the receptor protein on the surface of the target cell. That target protein, the receptor protein, then initiates a cascade of changes in proteins inside the cell that sets up a relay of the message to a transcription factor that will then enter the nucleus and activate transcription of the target gene that is the target of this transcription factor or transcription factors. That then produces the transcript, which is exported from the nucleus and gets translated by ribosomes into the protein that is being triggered by the presence of this signal protein, or whose presence is being triggered by the presence of this signal protein. Now, this internal relay system comes in several different forms. We're not going to talk in detail about the different forms of relay mechanisms that are involved. We just want to get an overall view of what is taking place as a consequence of the release of the signal molecule, its reception on the target cell, and the effect that has on the internal behavior, the biochemistry of the cell. So here is the initial part of the process. The signal molecule is released, binds to the receptor, triggers the relay that activates the transcription factor. That transcription factor then binds to the promoter region, or it may itself bind to the enhancer regions in the DNA if it's an activator protein, or it may interact with the activator proteins that are already bound to the enhancer sequences on the DNA. But the transcription factor serves to activate the particular gene that is the target, produces the messenger RNA, it gets translated, some of the pathways that are involved. So I forget how many there are here. Let me just double check. Okay, so there are 3 different pathways. So what I'm going to do is just put a note up here. So these animations can get a little bit detailed. I'm not going to hold you for understanding all of these relays, the relay mechanisms and the names of the molecules that are involved, because it's not important. These animations are included simply to illustrate different ways in which a message can get relayed from the receptor via different types of relay proteins to activate a transcription factor, which will in turn activate a gene inside the nucleus. So there are 3 different ways that are commonly referred to. You can look at these. Don't make too much of them. Don't try and learn all the intricate details of each of the pathways because it's not really necessary for you to know the intricate details of the pathways. But I think it's kind of cool for you to look at the animations and just get a feel for the different ways in which cells can regulate the expression of genes. So another quick break. Here we're not going to stop because we stopped just a couple of minutes ago, but another concept check here just to make sure that we understand what we've just been talking about. So how can a single molecule from one cell alter gene expression in the target cell without entering the target cell? Well, this is all about the signal transduction pathway. The binding of the signal molecule to the receptor on the surface of the cell triggers a relay mechanism inside the cell that activates a transcription factor that activates the genes. So maybe we can put something like that here. So the binding of the signal molecule to a receptor activates an internal signal relay that results in the activation of a transcription factor that activates the target gene. So the signal molecule itself does not have to enter the cell. All it has to do is trigger the internal signal relay pathway, and that will result in the activation of transcription inside the nucleus. So one of the things that we've learnt about the signaling pathways and the development of an organism is that there are key genes. We refer to them as master control genes or homeotic genes. You can see the name here, that don't actually code for structural genes or enzymes or anything like that. What these genes do is regulate whole groups of other genes. So you've got the master control gene at the top, and underneath you've got maybe hundreds of other genes that are activated when the master control gene gets activated, or a combination of master control genes gets activated. This is what I was hinting at when I was talking about the regulation of genes and the regulation of development, and the fact that humans only have a limited number of genes and not that many more than fruit flies, yet we are vastly more complicated than fruit flies. And the difference is that the number of master control genes that humans have is about 4 times the number of master control genes that fruit flies have. In terms of the other genes, we share a lot in common with fruit flies. After all, we all do cell respiration, we all do DNA copying, we all do cell division. There are lots of processes that we do in common with fruit flies. So not surprisingly, we share a lot of genes in common with them. But the one place where we differ is in terms of how those genes are regulated. Fruit flies have a small number of these homeotic genes, these master control genes. Humans have 4 times as many of these master control genes. And simply having that 4 times as many master control genes means that we can do so much more with the genes that we've got. A lot more than a fruit fly can do with the same set of genes, or nearly the same set of genes. And that's what makes us so complex compared to a fruit fly. In animals, these homeotic genes are called Hox genes. And the reason why I want to point this out to you is that we've found homeotic genes in fungi, we've found them in plants, we found them in animals, we found them across the board in all eukaryotic organisms. We have not found them in prokaryotic organisms, although there is a hint that maybe there are some precursor genes to the homeotic genes in bacteria, but there are no definitive homeotic genes in bacteria. The Hox genes that animals have are unique to animals, and so they are part of how we define animals. The presence of Hox genes is a defining feature of animals, but Hox genes are part of this larger family of homeotic genes. The reason why these are important is because these bind to DNA. They are DNA binding proteins. And in fact, one of the things that we notice about all of these homeotic genes is just how highly conserved, meaning how similar, are the regions that bind to the DNA across the board in these homeotic genes. There are very, very subtle differences, but not a huge difference. These are very, very highly conserved across the board. But that should not come as a surprise either, because these are the proteins that are going to bind to the regulatory regions of the DNA in a eukaryote that are in turn going to activate not one gene, but perhaps hundreds of genes in combination. So how significant can these Hox genes be? Well, here's an example of just how significant these Hox genes can be. So this is a photograph of two fruit flies. You can see on this fruit fly, so this is Drosophila. Fruit fly. So you can see that on this fruit fly, it's got a pair of antennae, which is what it should have sitting right there. This fruit fly doesn't have antennae, it's got legs. It's got legs growing where there should be antennae. Now, what is so remarkable about this is that the timing of when you follow through on this matters. So we know that spontaneously this can happen occasionally in fruit flies. This is a spontaneous mutation that gives rise to these legs where there should be antennae, because the signaling pathway was screwed up, right? Remember, what determines what cells become is determined by the signaling pathway and the signals that they get. And in this case, the signaling pathway got completely screwed up because of the mutation. But what is really remarkable about this is that in the laboratory, we can manipulate this system. If you look on the side of a developing fruit fly embryo, you can see little discs in what will become the thorax of the fly, the middle segment. And there are 3 of these little discs on each side of the thorax. That's where the legs are going to emerge from. If you take cells from those little discs and transplant them up into this region where the antennae are supposed to grow from, if you do it early on, very early on in the development of the fruit fly embryo, those cells will turn into antennae. They'll grow antennae. If, however, you wait 24 hours and do the same thing, those cells will grow into leg cells and you'll get legs growing. So you can actually create the legs by choosing when you move the cells from the little discs where the legs will eventually grow up onto the head where the antennae will ultimately grow. What we now know is that cells get signals telling them where they are in the developing embryo with respect to 3 axes. And the 3 axes you can see in your own body. So you have what is referred to as anterior to posterior, head to tail. Dorsal, which is back, to ventral, so back to front is typically how we say it, dorsal to ventral. And then left to right. Those are the 3 axes of our body. And the layout of our body is determined in part by the position of cells in the different structures that develop, like arms and legs. And they get information about where they are in 3 dimensions in those structures. And it's the Hox genes that provide that 3-dimensional information and tells the cells where they are with respect to those 3 axes. And when the cells finally get the signals that tell them where they are with respect to those 3 axes, they become committed to making the structures that are supposed to be produced in those locations. The term that we use for that is that we say that the fate of these cells is determined, and that these cells are determined. That's the fancy name that we give to this process. And so what's happened here, or what happens in the experiment that I was talking about, where you transfer the cells from the location where the legs are supposed to develop to the location where the antennae are supposed to develop. In the first situation, the cells in the little discs have not yet been told you're going to become leg cells. They've still not been determined. They haven't been told yet that they're supposed to become leg cells. So when you transfer them up to where the antennae are supposed to grow, they will eventually get the signal, well, you're going to become antennae cell because that's where you are. And they follow those instructions and they'll become antennae cells. If you wait that 24-hour period, however, in that 24-hour period, the cells in the disc finally get the instruction, well, you're going to become leg cells. And so their fate is determined. So when you transfer them up to where the antennae ought to be, because they've already received the instructions, become leg cells, they follow those instructions, even though they're now sitting where there should be antennae, but they're following the instructions that they've been given, and they're determined, their fate has been determined, and they grow into leg cells, components of the legs. So when we look at this checkpoint question, how can a mutation in just one homeotic gene drastically affect an organism's physical appearance? The key here is, remember that the Hox genes, or the homeotic genes, are not controlling a single outcome. They're controlling hundreds of outcomes by controlling hundreds of genes that sit underneath them. And so if you change the homeotic gene, and you change the way that homeotic gene is expressed, when it is expressed and where it is expressed, and in which combination of other homeotic genes it's expressed, you're going to change which genes are going to be controlled and turned on and turned off as a consequence of that. And that can create huge changes in the physical appearance of the organism. So here you have that spontaneous mutation that changed the antennae into legs. But once you recognize that that is possible, then you can start to get a better handle on the system by transferring leg cells from where they're supposed to be to where antennae are supposed to be, and seeing that the cells follow the instructions or don't follow the instructions depending on what instructions they've already received via the Hox genes. It's a very, very powerful system, and we're really only just beginning to understand it all. And there's so much that I would really like to tell you about this, because this is such a cool system. But we don't have the time, so we just need to move on, unfortunately. So one of the other things that has become really important in studying gene expression is to be able to tell which genes are being turned on in different tissues. Being able to say, well, this gene is on in this location, this gene is on in this location, and sometimes both genes are on in some locations. And so the researchers have developed this device called a microarray. It's basically a fancy microscope slide that's got a whole lot of little wells in it. And inside that well, you attach a piece of DNA to the surface of the well, and then you use a single strand of DNA that is complementary to the genes that might be there as a label to indicate which genes are active and which genes are not, because the single-stranded DNA will actually bind to the messenger RNA of the activated gene. and you use a colored tag on the single-stranded DNA probe so that you can identify where the genes are active, because the probe will stay stuck to the activated gene, whereas it'll get washed out if the gene is not active. And so you can tell where the genes are active by looking for the colors of the probes in these wells. So here's a picture of a DNA microarray. In this case, we've got two probes. One probe is red in color, one probe is green in color. And so inside each of these wells, you've got the target DNA being taken from different cells, and you're taking a look to see in which cells each of the genes that are being probed for are active. So in some of the wells, you see red. So that means that the probe for the gene, or the red probe, is finding the activated gene in those wells, and that's why the well glows green. In others, sorry, it glows red. In others, you can see that it's glowing green, and that means the other gene is active in those tissues, because the probe is binding to the activated gene. In some of the wells, you see yellow. So why do you see yellow? Because both of the genes are active there. The red probe is binding, the green probe is binding, and red and green gives you a yellow color in combination. So you can see that in many wells, both of the genes are active, telling you that those genes are active in that tissue. Where it is black simply means neither of the probes bound, so there's no active genes. Neither of the genes are active in that tissue. So you can very quickly screen huge numbers of cells. I mean, this could be cancer-derived tissue, it could be research tissue, but it allows you to screen hundreds of cells at once to find out whether genes are active in these cells or not. And so you start to get some clues as to what genes are being activated according to what sort of cells you're looking at. Whether you're looking at cancer cells or other types of cells, you can get a clue as to what genes are being activated in these cells by looking at these probes. It's a very, very quick way of being able to identify which genes are active in different types of cells. I put the concept check in here as well. I forgot I've transferred it here as well. Anyway, we've talked about this concept check, and I'm not going to stop. We can just keep moving, I guess. So maybe I should have stopped, I guess, but I'm going to keep moving. So now we're going to talk about something else that's involved gene expression. And it may not at first glance suggest that it's involved in gene expression. But let me assure you, gene expression and regulation of gene expression is at the heart of cloning, particularly when it comes to plant cell cloning, but also as we've been able to refine our techniques for cloning animals, we now know that part of the secret to being able to clone animals is also being able to change the expression of genes. So we've been able to clone plants for thousands of years. And if you've taken a cutting off a plant and put it in a pot and grown a new plant, you've cloned the plant. That is an example of plant cloning. And humans have been cloning plants in that fashion for thousands of years. But for about the last, let me see, the best part of 70 or 80 years now, we've actually been able to do something a little bit more significant in terms of cloning plants, because we can take pieces of tissue, small piece of a leaf, for instance, and by exposing it to different types of chemicals, we can cause the specialized plant cells to undergo a process called de-differentiation. Differentiation is when cells take on specialized roles. When that gets reversed, when they lose their specialized roles and take on a general role, then we say that they've become de-differentiated. And in terms of plant cells, it's relatively easy to do by exposing them to chemicals that are often referred to as plant hormones, but are more correctly referred to as plant growth substances. And different combinations of these growth substances can cause these plant cells that are already specialized to undergo this de-differentiation to give a ball of cells that we call a callus. I think I have a diagram of this. Yeah. So here's an example of a cloning experiment, and you might wonder, why clone orchids? Well, number one, this is a way of creating a lot of orchids very quickly, but number two, and I think I may have mentioned this to you before in a previous presentation, but getting orchid seeds to germinate is a really tricky thing to do. Only about 1 in 100 or so orchid seeds will actually germinate to give you a new orchid. So you don't have a very high rate of success. So if you want to make lots of orchids and you want orchids that are pretty much identical to each other, cloning them is the way to go. So when you buy an orchid at the nursery or from a truck on the side of the road, this is how they've been produced, through cloning. It's very easy to do. So you take a piece of the orchid tissue and you put it in the right medium, and the specialized cells in the orchid tissue lose their specialization. They de-differentiate into general plant cells. Then you transfer them to a new medium and they start to re-differentiate again, and they develop into all the parts of a plant. They actually develop into a little plantlet, and you may get several plantlets. You may get 10 or 20 little plantlets, depending on the size of this mass of cells, the callus that develops from these unspecialized cells. You can then cut out each of those little plantlets and put them into some more growth media with some more of these growth substances to support them. They will then grow roots and shoots and develop into a true little plant, which you can transplant into a pot, and that will develop into the orchid that you're going to sell at the nursery or sell from the truck at the side of the road. This is a very easy process to do. And lots of different types of plants are able to be cloned in this fashion. In fact, most plants can be cloned in this fashion if you wanted to go ahead and do this sort of thing. But commercially, orchids are really cloned a lot because it is so difficult to grow them from seeds. As I said, about 1 in 100 orchid seeds actually will germinate. It's a very tricky thing to do. But it is the only way to develop a new orchid. But once you get that new orchid, if you want to make many copies of it, this is the way that you would do it. It's very easy to do. I know people who do this on their kitchen bench. It's that easy to do, as long as you can maintain relatively sterile conditions, and that's not terribly difficult to do either, then you can do this on your kitchen bench. It's very easy to do. So where have we used plant cloning? Well, have you ever eaten a navel orange, or a seedless watermelon, or seedless grapes, or any other plant fruit that is seedless? Obviously, if they don't have seeds, you can't grow them from seeds. So how else are you going to grow them? You grow them by cloning them. So in the case of oranges and grapes, I can tell you how that's done. I'm not entirely sure how they do seedless watermelons, but I know how they do seedless grapes and seedless oranges, the navel oranges. And in fact, all navel oranges around the world are derived from a single tree that was found in the backyard of a family living in the town of Redlands in Southern California. It's east of Los Angeles. I've actually seen the original navel orange tree. I had the good fortune to live in that area for a number of years when I first came to the United States, and I was able to see the original navel orange tree. It's still alive. But all navel oranges are derived as cuttings from that tree. And the way you do it is to grow what's called a rootstock tree. So this is a tree that is got very strong roots and will grow in under the conditions that you want to grow the tree. Once you get it big enough, you cut a notch in it, and you take a branch from the navel orange and you notch it and fit it into the notch that you've cut into the rootstock tree, and you bind it up and you just leave it. And over time, the two bind together and connect up, and the navel orange branch becomes part of the tree. You then cut off everything that you don't want, leaving behind just the navel orange part of the tree, and it will develop into the full tree with all the branches and everything and produce navel oranges. And basically for navel oranges, seedless oranges, and for seedless grapes, that's how you propagate them. That's how you produce more of them. You take branches off existing grapevines or existing navel orange trees, you notch them, put them or you create a wedge on the edge of the branch, and you put that into the notch of the little tree or the little vine that you want them to become part of. You bind them up, and they make special materials for binding them up, and you just leave them attached, and the tree does the rest, or the vine does the rest. They grow together and merge together to become one, and you take off all the extraneous material that you don't want, that's not going to produce the oranges or the grapes that you want, and now you've propagated it. You've created a new seedless grapevine or a new seedless orange tree. So if you buy a navel orange tree from a nursery, or if you've got a navel orange tree growing in your backyard, now you know where it came from. Again, I don't know how they do seedless watermelons. I ought to find out sometime. But that's how they do seedless grapes and seedless oranges, or navel oranges. One of the other things that is sort of a kind of cloning is when we have animals that shed parts of their body. So you know that some lizards can drop their tail when they're threatened by a predator. The predator goes after the tail because it wriggles around, and that lets the lizard run away. Salamanders can do a similar sort of thing. There are other animals that live in the ocean that can excrete parts of their body, or if you've got a sea star, for instance, you cut it up into pieces. So long as the piece has a part of the central disc, it can replace all the missing pieces. And in fact, abalone fishermen in California thought they were doing themselves a favor by cutting up sea stars, because they figured the sea stars were competing with them for the abalones. And so they would take the sharp knives that they used to harvest the abalones and cut the sea stars up into pieces and throw them back overboard, thinking, well, I just killed off a sea star. Well, if they cut the arms up and left a piece of the central disc on the arm, it would simply grow into a new sea star. So rather than killing off the sea stars, they were actually adding new sea stars to the population because they would survive. This is called regeneration. Lots of animals can do this. What is really interesting here is that the genes that are involved in this regeneration are found fairly uniformly across a lot of the animal kingdom. We actually have these genes. However, they are permanently disabled in us so that we can't regenerate missing parts. But there are researchers looking at trying to find ways of regulating these genes and figuring out how to turn them back on again. Because then if you lost a finger or something like that, you wouldn't have to salvage the finger and run through getting it reattached and hoping that it all worked again. You could simply grow a new one by reactivating the genes and regenerating a new finger. I don't know how far out that work is, how far along they are with that work, but I know that there are people working on it. We just don't have the ability to regenerate the missing pieces, but other animals certainly do. And as far as we can tell, we do possess all the genes that they possess for the regeneration. They're just not active in us. So one day, maybe in the future, we will be able to grow a missing finger or a missing toe or something like that. It'd be kind of cool, I guess. In terms of cloning animals, however, in the most common way in which animals are now cloned, if animals are cloned, and there's a lot of ifs involved in that, is through a type of cloning called reproductive cloning. In reproductive cloning, an egg cell, which is already primed to divide, has its nucleus removed, and then the nucleus from the host organism is transplanted in, and that host cell will have a nucleus that contains a complete set of chromosomes. So it will be a diploid set of chromosomes. So when the egg cell is then activated to divide, it simply undergoes mitosis and starts producing cells. But these cells coming from an egg cell retain the ability to then become all of the different types of cells in the organism that the embryo is going to develop into. This has been the gold standard for cloning animals, and it's taken a long time to get to the point where we're able to do it. So the note here says, and the textbook points this out, that Dolly, the first mammal to be cloned using a nucleus from a non-reproductive cell, was generated in 1996. Now, unfortunately, Dolly had a number of health issues as a consequence of being a clone, and this is one of the reasons why there is a moratorium right now on cloning humans, because of part of the issues that we know from cloning other animals, like Dolly as a sheep. So one of the issues that we know is associated with cloning is that an individual's DNA has an age attached to it. And when you clone an organism using the DNA from another organism, the clone has a genetic age the same as the donor organism. So when Dolly was cloned, while she developed as an embryo in the body of her surrogate mother and was born as a lamb, genetically, her DNA was as old as the donor sheep that provided the nucleus from which Dolly was cloned. So Dolly was already genetically old when she was born. That was part of the problem. The other problem is that cloning in animals also appears to give rise to a number of follow-up health issues. They're never quite as healthy as the original organism. And Dolly was actually euthanized because of some of the health issues that she had. Now, she is not fully departed because they took Dolly's remains and took her to a taxidermist who stuffed and mounted her, and she's now on display in the Science Museum in London. And I've actually seen Dolly because I've been to the museum, and Dolly is there, and you can see her. She's in the foyer of the museum, along with the first model of DNA produced by Watson and Crick, their first full-scale model. A number of other things in the museum. It's a fascinating museum to go and visit if you're ever in London. But this was part of the problem. The other thing is that there's not a high rate of success in cloning animals. Dolly was actually number 269 of the attempts that were made to try and clone a sheep. There were 268 failures before they got to Dolly. And again, this is part of the reason why we do not consider cloning humans to be something that we ought to be doing right now. There are too many things that can go wrong. There are too many things, too many problems yet to be overcome to make it worthwhile cloning a human. Then if you want to add the final piece to the puzzle, if you recall back to when we were talking about meiosis, and we talked about the number of possibilities, genetic possibilities there were for your parents when they had you. You were one of about 64 trillion possibilities. And we talked about the fact that you are genetically unique, but in fact, when you layer on the idea that we are not just the product of our genes, but we are the product of our environment as well, any clone that we were to make of you might share your genes, but they certainly won't have experienced your life, and so they won't exactly be you. Now, a few years ago, I think it's probably about 15, 20 years ago now, there was a company in Korea that offered to clone people's pets. I believe the company has now gone out of business. But for a while there, they used to have people sending them tissue from their dog that was getting old, for instance, and they wanted to clone so that they could continue to have a relationship with their dearly loved dog. The problem was that what they were getting was not what they thought they were getting, because the dog that they got, number one, was genetically as old as the dog that was being cloned, which didn't help very much. And number two, because you're not the product of your genes, you're the product of your genes and your environment working together, the dog that they had was not the dog that they loved. It was a different dog. It may have looked the same, but it was not the same as the dog that they wanted. And it was costing a lot of money. You could pay, I think the fee was somewhere around $100,000 to get the clone made. Kind of expensive. So where do we use reproductive cloning? Is there a purpose for reproductive cloning? And this just runs you through the technique of nuclear replacement cloning, which is what gave rise to Dolly. So you can take a look at that diagram. Is there a purpose for reproductive cloning? And the answer is, yeah, maybe. So in some cases, it's been used to clone animals that had traits that were valuable. So one of the very early cloning procedures or processes that was carried out after the cloning of Dolly was to take tissue from a cow that produced a lot of really high quality milk and a high volume of quality milk, and try and clone that cow to produce a herd that had those characteristics. I'm not sure what happened to that experiment because very little has been said about it. I have a feeling that a lot of these ideas sounded good on paper, but in terms of execution, not so much. There has been work done in the pharmaceutical industry, and I'll just show you this picture, I guess, of cloning pigs with a human immune system. And you might ask, why do we want to clone pigs with a human immune system? Well, one of the issues that we have, as far as humans are concerned, is having enough transplantable organs. People can wait on the transplant list and die waiting for an organ because they can't get a good match between a donor and a recipient. But it turns out that adult pig organs are about the same size as adult human organs. And so long as the pig has a human immune system, you reduce the chance of the organs being rejected. And you might go, oh, that sounds disgusting. Why would you want to put pig organs in a human? Well, just remember that one of the first trans-species transplants was transplanting a baboon heart into the body of a baby who had a failing heart. I actually worked at the university where that was done, Loma Linda University in California, where the first baboon's heart transplant into a human baby took place. If this was to provide an available source of transplantable organs that were compatible with humans, it would take some of the pressure off the transplant system. Right now, it's not a viable method, but there are companies that are continuing to research this. But there are a number of other ways in which we may get around this in the future, like growing organs in the laboratory. Yes, I said that, growing organs in the laboratory. It can be done. We can do it with rat organs right now, and they are perfectly functional. And we can do it with pig organs, and there's nothing to say that we could not do it with human organs too. We could grow human organs in the laboratory. or in a organ factory, so to speak. Probably a few years down the road yet, but the technology does exist to be able to do that. It's just making it work satisfactorily and reliably in human situations. There's also been attempts to try and clone endangered animals. So you can see two relatives of cattle that have been cloned. Unfortunately, the clones did not survive for the same reason that Dolly had to be euthanized, because of health issues. So whether this is actually going to be viable or not in the future remains to be seen. But these have been some suggestions that have been raised and some ideas that have been tried to make use of our ability to produce animals through cloning. There is a second type of cloning that is available as well, and that's called therapeutic cloning. Not cloning for the purpose of creating an organism, an adult organism, but creating an embryo that contains embryonic stem cells. So what is a stem cell? A stem cell is a cell that can develop into a range of different types of cells. An embryonic stem cell is a cell that can become any of the 220 different types of cells in a human body, for instance. So a human embryonic stem cell has the capacity to develop into any one of the 220 different types of cells that are present in our body. They are the gold standard by which stem cells are measured, because they have that capacity. The term that we use for them is to say that they are totipotent. Let's see if we can do this. Totipotent can become any cell type in the body. Now, there are also another type of stem cell called adult stem cells, and let me put the note down here. These are said to be pluripotent, which means they can become some of the cell types. In the body. In other words, they have a limited range that they can become, whereas the embryonic stem cells can become anything. Now, we have a reasonable understanding of how to get stem cells to develop into different types of cells. It's all about exposure to different types of signaling molecules. And as we learn more about the role of the Hox genes, we are learning more about what signaling molecules are essential for taking stem cells in different directions. Basically, what you have, if you're thinking about the 220 different types of cells in your body, it's kind of like a tree with many, many branches. And so at the base of the tree is the embryonic stem cell, which can become anything. And then you have a couple of big branches from which smaller branches extend, and out on those bigger branches and the smaller branches are the different types of cells that are present in the body. And some cells and the relationships between cells are more or less starting to be understood in terms of what sort of stem cells they come from. We all have these adult stem cells in our body. The problem is that they're buried in amongst lots of other cells, and it's very difficult to recognize which are the adult stem cells from the other cells, because they're all mixed up together. But all of our organ systems have some of these adult stem cells in them for the purpose of repair and replacement. If we can figure out a way to successfully use embryonic stem cells, then we may be able to repair some types of tissue damage. So for instance, if you get spinal damage, that can be permanent because nerve cells do not replace themselves. They do not grow and divide, they do not replace themselves. If they get damaged, it's a permanent damage. But there is a suggestion that we may be able to use embryonic stem cells to repair spinal damage, for instance, because there have been some suggestion that this can be done in rats using embryonic stem cells. There are a couple of studies that have been approved to look at the use of embryonic stem cells to fix some of these issues. In Europe, they've been doing this for a while. Here in the United States, not so much. It's only been in about the last 8 to 10 years that approval has been given to actually work with embryonic stem cells to look at ways of curing some of these tissue damage conditions. There is perhaps another solution, however, and that other solution is to start with cells that you might think have no chances of becoming embryonic stem cells or becoming stem cells at all. Because research is now being done with skin cells, and we can grow skin cells relatively easily in the laboratory. They're fairly easy to grow. And it turns out that to take a skin cell and turn it into an embryonic stem cell, which at first glance sounds like something that might be really more or less impossible to do, is actually not that difficult to do. It turns out that there are 6 genes that need to be reactivated in a skin cell to turn it into a stem cell. And about 10 years ago, the first papers were published that identified those cells and provided data that showed how a skin cell could be turned into an embryonic stem cell by inserting in active forms of these 6 genes into skin cells. It's not an easy thing to do, but it can be done. And then you get the embryonic stem cells. And then you can take those embryonic stem cells, and because we know a little bit about how these cells develop following the different branches on the tree, you can start to use them to create different types of cells that might be useful for doing tissue repair. So if we look at the adult stem cells that are found in bone marrow, we know that those can become all the components of blood. So red blood cells and white blood cells that are components of the immune system. They also have the capacity to turn into nerve cells, and they also have the capacity to turn into heart muscle cells. In fact, if you can isolate some of these adult stem cells from the bone marrow and inject them into a damaged heart, they can repair the heart. This has been done in rats. So we know this technology, or at least the idea has merit because we can do it in rats. It's not been done in humans yet. But we also know that embryonic stem cells can follow the same pathway, these separate pathways. So it may be at some point down the road, again, how long down the road, I don't know, but somewhere down the road, it may be that if you suffered a mild heart attack that didn't wind up killing you or something like that, that doctors may be able to take a small amount of your skin, add in the appropriate genes, turn them into embryonic stem cells, grow out of those some cells that could be then injected into the damaged tissue of your heart, and the damage repaired by those cells that were derived from you. So you wouldn't need to transplant a heart. You simply fix the damaged heart. That's the sort of possibility that is being talked about. The problem is that every time someone says, well, in about 10 years, it never comes to fruition. The 10 years just keeps getting 10 years further apart from where we're at. Whether we will ever truly be able to make use of stem cells in the fashion that people have talked about them remains to be seen. I am keeping my mind open on this one. I've heard lots of promises. I haven't seen lots of delivery on those promises yet. But if in the future we are able to deliver on those promises, then stem cells could be a very, very exciting part of medicine in the future. Then there is another source of stem cells that remains a relatively untapped source of stem cells, and that is fetal blood taken from the umbilical cord at birth. This is being actively researched. There does seem to be some merit in taking these stem cells. They may actually be a potential source of therapy in the future, but for now, like all stem cell research, it's still basically built on promises. But as you can see from the note here, pediatricians are recommending that blood be banked for families where there is a genetic risk, because in the future we may be actually able to deliver on the promise of stem cells. And it would be a shame if you didn't have the stem cells available to be able to utilize. So for families where there is a significant genetic issue, banking the umbilical cord blood is being recommended by pediatricians, but not for anyone else, because we don't know if it'll work or not. So I've talked about adult stem cells. We can move on from there. So let's take a quick break here, and this will be a full break. I'll stop the recording and meet you on the other side of the break. So how do the results of reproductive cloning and therapeutic cloning differ? See you on the other side of the break with the answer.